Hereby, I open this academic ceremony in which Sinead Greally will defend the academic thesis, Systems Biology Analysis of Synucleinoptathies. May I invite you to present a summary of your study and the conclusions of your thesis.
My PhD thesis is titled Systems Biology Analysis of Synucleinoptathies. So what are synucleinopathies? Well, over 200 years ago in 1817, James Parkinson first described what he called shaking palsy in patients that showed tremors and rigidity. And since then, two other similar disorders have been described: DLB, or dementia with Lewy bodies, and multiple system atrophy, or MSA. PD, DLB, and MSA were all termed synucleinopathies in 1998. So Parkinson's disease, dementia with Lewy bodies, and multiple system atrophy are all called synucleinopathies. However, they have very different clinical symptoms and manifestations. PD is the most well-known out of the three, and it's actually the second most common neurodegenerative disease after Alzheimer's. DLB then is the next most common synucleinopathy, and is actually the second most common type of dementia. Lastly, MSA is actually termed a rare disease due to its low prevalence. And all three of these diseases are termed synucleinopathies because they're believed to be caused by a protein, alpha-synuclein. And we see that in the brains of these patients post-mortem. Alpha-synuclein forms clumps that we see in patients with PD, DLB, and MSA. They form mainly in the midbrain of patients with Parkinson's, in the frontal region of the brain in patients with DLB, and in the back of the brain in patients with MSA. But why or how does alpha-synuclein aggregate? Well, the answer is we still do not know. We all have alpha-synuclein in our brains. However, in these patients, under some kind of pathological conditions, the alpha-synuclein begins to clump and aggregate together to form aggregates called Lewy bodies. Once inside the cells of the brains or neurons, these Lewy bodies have detrimental effects, causing a lot of stress, which will inevitably cause the cells to die in the brain, where we see the degeneration in patients with synucleinopathy. So why are we researching these seemingly complex diseases? Well, apart from the patients themselves, synucleinopathies have a huge economic burden. For example, Parkinson's disease alone costs billions of dollars in the US in disability income and medical needs. But apart from that, the main focus for research is, of course, the patient and the families themselves. And there are three main aspects which we want to research for the synucleinopathy patient. One being drug therapeutics. Currently, the only way to manage synucleinopathies is purely to treat and manage the symptoms. There is no cure, and on the market today, there is currently no disease-modifying therapies, so no drugs that can slow down the progression of synucleinopathy. The next aspect in which we want to research is diagnostics. It would be better, of course, if we could get an early diagnosis of these diseases. However, there are some challenges, like overlapping clinical symptoms between patients with PD, DLB, and MSA, and of course, other neurological disorders like Alzheimer's. There is also a lack of biomarkers. A biomarker is something we can measure in something like the blood that can distinguish and diagnose a patient with PD, DLB, and MSA. And lastly, in order to actually get a definite diagnosis, we need to look at the brain post-mortem, like I showed earlier. And the last aspect in which we want to research these diseases is to understand the disease mechanism. Why or how does alpha-synuclein aggregate? Not only that, but what other proteins and pathways and molecules are being affected here, and could they have a cause? And what is the overall dysfunction in the cells of the brain? If we are able to further understand the disease mechanisms, this will in turn will be able to help develop drug therapeutics and, of course, diagnostic markers. One way in which we can research synucleinopathies is using proteomics. Proteomics is defined as a large-scale study of all proteins made up of a cell, a tissue sample, or an organism to understand their roles, interactions, and therefore their impact on health and disease. So I show here a simplified workflow for mass spectrometry-based proteomics. So if I have a sample of Parkinson's disease human brain tissue, the first thing I would do is extract the proteins. I would digest them or break them up into smaller fragments called peptides, and using liquid chromatography, run them on instruments known as mass spectrometers. These are very, very powerful and sensitive tools that, using the mass over charge ratio, can actually identify what proteins make up that sample. We then can carry out analysis, compare that Parkinson's sample to another disease or even to healthy controls. And this is exactly what we did for my PhD thesis. For my first study, I carried out a first-of-its-kind comparative proteomic analysis, where I compared human brain tissue samples from a cohort of controls, therefore patients who did not have any pathology, Parkinson's disease patients, DLB patients, and MSA patients. I used cortical human brain tissue and ran them on the mass spectrometer. From this alone, I was able to identify 3,000 proteins in just a small human brain sample, and then I could compare them to each other and to the controls. So visually shown here as a volcano plot, for my patients with DLB or dementia with Lewy bodies, when I compare them to the healthy aged controls, I can see that there is significant dysregulation. Shown in red is some of the upregulated or increased in expression proteins I identified, and shown in blue are the decreased in expression proteins when I compare them to our healthy aged controls. We did the same thing again with our Parkinson's disease tissues. We can see again significantly up and down regulated proteins when we compare them to controls. However, straight away we can see there are some similarities to the DLB patients, but also some differences already emerging. And lastly, we also compared our multiple system atrophy samples to our controls. And what we found was, once again, a lot of increased, shown here in purple, proteins, and decreased shown in blue. Once again, we can see some similarities between MSA, PD, and DLB. However, we can see there is once again a lot of differences also. The power of using mass spectrometry means we can identify some interesting proteins, some shown here that have already been associated with synucleinopathies. Amyloid beta, Huntington, sequestosome were all increased in all three diseases. And proteins like albumin, paravalbumin, and even PARC7, a well-known genetic risk factor, were all decreased. However, the real power of mass spectrometry is identifying new proteins that have not been previously associated with the disease. And we identified them. Examples include annexin A7, colin-2, and MYADM we found were all increased in all three diseases and had not been previously associated with all three synucleinopathies. We also found some interesting proteins like THOP1. This is a protein that actually breaks down aggregates. We found that it was decreased in all three diseases, and therefore something like THOP1 could be a potential drug target for the future. So from this initial analysis, we could create a summary figure of some of our findings between DLB, PD, and MSA. Here highlighting some of the common proteins dysregulated in all three, some new and some already associated, but also identifying some interesting proteins that haven't been associated previously, or proteins that were only dysregulated in one or two synucleinopathies and not all three, further showing that there is a big heterogeneity between the three diseases, despite all being believed to be caused by one single protein. For my next study, we then focused in on some proteins of interest. Here shown is our DLB patients. We identified the protein tau was upregulated exclusively in this cohort. Tau forms fibrils and tangles in patients with Alzheimer's. So because we have the commonality of dementia here, it did make sense. However, when we looked further, we found that this upregulation of tau was actually driven by 9 patients out of the 30. The other 21 patients showed similar levels of tau as that to the controls. Based on this, I stratified my DLB patients into DLB tau positive and DLB tau negative. And looking at the tissue, it confirmed these findings. In a patient deemed DLB tau negative, we found that tau did not show any aggregates. However, in a patient termed DLB tau positive, I found aggregates of both tau and alpha-synuclein. And this led us to question, is neurodegeneration more of a spectrum? Instead of classifying these diseases as synucleinopathies or tauopathies, is it rather a spectrum or a continuum of the protein aggregate-prone proteins? If we are able to classify patients along this spectrum, it would improve diagnosis and therefore prognosis, and also give way for more personalized therapeutics based on these 2 proteins. As recently, more emergence of drugs targeting alpha-synuclein in particular, or tau, have been coming onto the field, and therefore patients that fall along this spectrum may benefit from both types of therapeutics. Lastly, we looked at another interesting protein or pathway of interest in MSA. We identified STAT1 was significantly upregulated in the patients with MSA. STAT1 causes inflammation. Inflammation is what happens when our body is fighting a cold or a pathogen, and inflammation in the brain is something that has been observed in patients with neurodegeneration. So I wanted to investigate the exact effect of inflammation on the aggregation of synuclein. For this, I created a cell model. I transfected cerebellum tissue from MSA patients and controls into cells overexpressing synuclein, and I added cytokines interferon gamma and TNF-alpha to cause the inflammation. What I found was, with just the seeds alone from the MSA tissue, we began to see this clumping or aggregates of alpha-synuclein. However, when we added inflammation, these clumps or aggregates of synuclein were everywhere, clearly showing that inflammation has a direct effect and increases the aggregation of synuclein, once again showing that inflammation could be a possible drug target, in particular for MSA patients. So overall, I believe my PhD thesis aided in our understanding of neurodegeneration, in particular synucleinopathies. My first study, a first of its kind, comparing all 3, found both similar, but also more importantly, unique protein signatures between PD-DLB and MSA. My next study showed that there could be a potential spectrum of neurodegenerative diseases, in particular using synuclein and tau. And lastly, we observed the direct impact of inflammation on alpha-synuclein aggregation. We believe these could have clinical potential as we've identified new targets for potential drugs or diagnostics. However, the next steps would be to further investigate any of these findings and to validate them. Thank you.
Thank you very much for that very clear presentation. The opposition will now be opened by Professor Maarten Honing, who has a chair in Analytics and System Imaging at this university.
Thank you, Mr. Rector. Dear candidate, first of all, congratulations with this, I would call it, thorough way of working and nice research, and also the supervisors for this achievement. I think it was, and it has, quite some important steps to fill this whole of unknown field of this neurodegeneration. And I would like to ask one of your assistants to read proposition number 9.
You don't have to see the whole staircase, just take the first step. Martin Luther Jr.
Thank you very much. So, dear candidate, a little bit to tease you, is this not an excuse for you to start with some research only in small steps instead of just asking the big question, is alpha-synuclein really the central dominator of these 3 diseases? Because Would you not already know the answer?
Thank you, highly esteemed opponent, for that question. So when I saw this quote initially, it really had a visceral reaction to me. For me, it really described my PhD thesis journey. And I think it's actually a really important quote for science in general. I think...
to have faith in yourself or just do small steps?
I think...
And lose the picture?
No. I think the small steps for me is following the data. And instead of starting with a question...
But I hope you agree that science is about hypothesis.
Yes.
And asking you the big question and then to see how you can give an answer, even in a small step.
Yes, I agree. So starting with a hypothesis and a driven idea, for instance, are the synucleinopathies different? Do they differ on a proteomic level? The small steps in that case are following the data and not...
As a chemist, like myself, I would say, per definition, that should be the case. Otherwise,
Yes.
I don't have 3 different diseases.
Yes.
Okay. No, okay. So it's about faith, it's about your journey, but we agree the big picture is very important to research.
Yes, I think the big picture is very important. I think in particular, knowing what your data can tell not only you, not only your lab, but the field itself.
Let's discuss that later, because there I have some questions for sure. And I already start with an intriguing question, because biology and molecular interactions is nothing else than interactions of 3-dimensional structures of molecules. So bioactivity of a molecule is defined by its structure and not by its amino acid sequence, which you are analyzing with your MS. And you even state that the folding is important. And I suppose that the folding is also highly dependent on the change of the alpha helix of the N-terminal part, where most of the acetylation post-translation modification takes place. Now I'm going to accuse you from something, because you were very rich with a timsTOF mass spectrometer. So why are you not analyzing the folding of the molecule in just study just the amino acid sequence with this beautiful eye mobility?
So for this initial study, the focus, of course, was the alpha-synuclein itself. We worked on optimizing sarcosyl fractionation using sp3 in a tryptic digest. And using the timsTOF, we were able to get a beautiful sequence coverage of the whole...
How much? About 100%?
About 87%.
Oh, yeah, that's typical biochemical approach.
Yes.
So 80 is 100% in your... But we were speaking about folding.
Yes,
I'm not interested in the sequence.
Yeah, so I do agree that using mass spectrometry, we cannot exactly assess the folding or the, cryo-EM structure of the synuclein itself.
Because you agree that this aggregation is highly dependent on the change of the middle part of the molecule.
Yes.
And then structural information is much more important than all the other information.
I think what is important using the mass spectrometry data in particular is we were able to see areas of modification, so sites of modification, acetylation, ubiquitination, phosphorylation, in addition to cleavage sites. So areas where we see, a cleavage or a truncation, which then in turn can give us more information on the cryo-EM structure.
Absolutely. I saw also that especially post-translation modifications on the N-terminus has an effect on the clearance via the ubiquinine system. So rightful taken. But I would like to understand how the transacetylation changes the 3-dimensional structure of the alpha helix. But I think that's maybe for later to have a thorough and heavy discussion on that. I would like to go into some definitions, and especially, let me see my paper. On page number 20, you speak about mass spectrometry, and I call myself a physical mass spectrometrist, and you are very positive. And then you say you use the bottom up because the other, let's say top down, does not allow for high throughput. And then I look to all your essays, and a just simple essay, in your case, takes more than 2 hours nearly. So how high throughput is your method compared to the other method?
So,
and how important is high throughput? Why should we want high throughput?
So I think it's important for high throughput, in particular, when you're assessing the human brain tissues. We want as large of a cohort and patient sample set as possible. And
But why? Why? Do we need it tomorrow, the data? Or do we want to have information?
No, we want to have information. I think based on statistical power and everything, we want to have as many patient samples as we can possibly look at. In terms of using the timsTOF, we used, yes, a gradient of 90 minutes for this initial study using DDA. And the reason for that is because it was an initial discovery study. So we did not...
maybe we can go on that, because you used formic acid 0.1, and I don't like formic acid, it's oxidative reagent. So I would say half of your proteins are not the proteins you thought they were there. And did you validate that anyway? But that means also that the pH is 1, and then you use a C18 column.
Yes.
Like everyone, I suppose. But why not a HeLiC or a cyano column, who are nowadays used in two-dimensional LC?
At the time when this was first ran, this initial study, we did try different filters, and we found the C18 column gave
filters, the LC column, I mean.
No, for the C18 columns, I mean, sorry. We did find that this gave us the best coverage for the synuclein, which was our initial target that we could see.
Interesting. Yeah.
Yes.
Okay. So the C18 was doing the job, 25 centimeters long and 2 hours run.
Yes.
To get quickly data on the brain.
Yes.
Okay, that's fine. Okay, that's also maybe another way of speaking about it. Maybe we go to the same page or the page before, and you use a lot of beautiful words, and maybe we can discuss as scientists that sometimes they are a little bit too heavy. Unprecedented levels. Now, that's a lot. And is that really true with mass spectrometry?
I do believe, based on our analysis alone, and even since this initial analysis with the increase in new methods, more targeted methods, and more sensitive mass spectrometers, I think it is unprecedented levels.
Now, for me, fluorescence is not to beat, theoretically not, so maybe you can have a discussion that unprecedented is very good levels. That is fine with me. And also remarkable precision. Oh, that is a dangerous one. What's remarkable? Because that seems that you discovered something which you did not expect from the beginning. What do you mean with remarkable precision? Is that good or bad?
I think any type of precision in terms of data analysis is obviously important. You want to know what you're identifying and seeing is precise and accurate.
Yeah, that's what exactly, because I normally criticize the proteomics people a lot, because sometimes biological variation is less than 1%, and your mass spectrometry will never pick up changes in intensity of less than 10%. So maybe you are looking to something and you cannot find. So this remarkable precision is not that remarkable then.
Okay, maybe we can get there with more targeted analysis.
Okay. Another one, and that is nice, and I think you already said it in your presentation, I like it a lot. It's, let's say, the opportunity to find new proteins, new targets, and hopefully also to find new therapeutic drugs. And you use here in your thesis even the statement that mass spectrometry will lead us to interesting targets for drug treatment. Is mass spectrometry doing that, or are there other rules for a protein to be druggable anyway?
I think the power with mass spectrometry is being able to identify a large set or number of proteins, which would not be possible using some other methods. I think, therefore, we are able to identify a large number of potential proteins that are of interest that could possibly be druggable or biomarkers, etc. But of course, that needs further investigation.
The requirements for druggable are completely
I think if a protein
different from what a mass spec can give you.
I think if you see my third study where I created the cell model, you can see there I investigated and saw the direct effect of STAT1 activation.
Then I'm very happy that you were doing that, and I give the word back to the rector.
Thank you very much to both of you for this very nice warming up, but we have to continue indeed with Professor Theo de Kok, who holds a chair in population-based toxicogenomics at this university.
Thank you. Dear candidates, my compliments for your thesis. It's very well written, very clearly structured, and it has an exceptionally nice introduction, a remarkably nice introduction maybe. Because I'm from the domain of toxicology, and so some of these things that you have been studying, the disease phenotypes, are new to me. So I learned a lot, so thank you for that. I would like to go now to chapter 2, where you present the proteome landscape of 3 different diseases that you have explained, and in supplementary table 2, you present an insight into the data quality check. And your conclusion on page 40 is that there's actually no bias as a consequence of gender, age, but also batch. And that triggered to me the question, how do you define a batch?
So thank you, highly esteemed opponent, for that nice words and question. So in order for this study to be ran, we actually randomized the 4 groups into batches, 8 batches in total with equal amounts of PD, DLB, and MSA and controls. And they were randomized and ran on the prepared and ran on the TIMS TOF based on these batches. So that was they're referring to the batch.
Okay, so I don't know if you have the thesis in front of you, but if you go to this supplementary table, I think I see some, so typically batch number 2
Yes.
shows some tendency to, in number C, that it deviates away from the rest. So my question is, how do you then determine that there is no such impact? So for the rest, I'm pretty much okay, but that is something that stood out to me. And my next question was, how did you randomize them? But you explained that already.
Yes, thank you for that question. So I can see what you're saying, that you can see slightly some of the batch 2 do group together in that corner.
They, to me, I would say they cluster quite together, deviant in principal component number 2, away from the rest.
Yes.
So what is your? So how do you come to your conclusion that there's no such impact of batches?
So, based on the whole 8 batches, we believe that there was no overall batch effect.
I believe we already discussed religion and what we believe. I'm asking for a strict criteria here.
I do think that overall there was no batch effect with the total 8 sets of batches. However, I do acknowledge that there are several batch number 2 samples.
That's what I wanted to hear. So you believe me also a little bit that there might be a batch effect, and then you could have said that principal component number 2 only explains 9% of the variance, which is limited. So the impact, I guess, would be limited.
Yes.
So going on to the methods of chapter 2, I had some confusion about what is the definition of differentially expressed or significantly dysregulated, because you describe 2 different FDR corrections, one of 1%, which is relatively high, and one of 0.05. Can you explain that?
So for the initial identification of all proteins and peptides using MS-Fragger, we had an FDR of 1% for both peptides and proteins. For the differential analysis, comparing controls to the disease group, I used a standard student's t-test with an FDR of 0.05% for that analysis.
Okay, and then I see a remarkable finding that for MSA, which is the smallest group, half of the size of the rest, you have the highest number of significantly dysregulated proteins, over several hundreds actually, to be honest. And then if I look at Parkinson's disease group, for instance, they had the lowest number, and none of that survived this FDR. What does that mean?
Yeah, so that was an interesting find for sure.
Interesting, meaning something you didn't expect? Or?
I would say slightly unexpected. Because MSA is termed a rare disease, there are not a lot of proteomic analysis on patients with MSA. Therefore, we did not have a lot of references for these analysis. We saw that they had actually...
No, but I'm not asking for the references. I think my question is could there be other explanations why typically this disease phenotype shows the strongest response?
So out of the 3 synucleinopathies, MSA is the most progressive out of the 3. It is believed to be the most detrimental with, I think, 6 to 10 years after diagnosis versus PD and DLB, where a patient can live up to 20 years with the diagnosis. Therefore,
that's remarkable, because because my line of thinking was that maybe these patients survive longer and have longer times of some phenotypes to build up a certain signature, and then what you study in this proteome landscape would actually be a very poor predictor of early onset at the earlier stages.
Yes, so they actually are the most aggressive form of the 3 versus PD and DLB. So we believe that the significant proteomic changes observed may be due to the more aggressive form of synuclein pathology observed in these patients. The other aspect is also a lot of MSA patients, the initial aggregation occurs in the cerebellum and then progresses to the cortex, therefore showing that we may have more regional differences here versus the patients with DLB and PD.
Okay, so I'm happy with that response. So you find a lot of dysregulated proteins, some are in processes which make a lot of sense, which is always good and nice. You also identify some new proteins, but the key question is, of course, the ones that can discriminate. And then I read on page 52, your strategy is that you aim to identify dysregulation in 1 or 2 groups. Why not just one by one? So if you look at figure 5.A, you have a Venn diagram and showed actually that you have unique proteins for all groups. So why not first dive into those?
So with the data set this size, we had a lot of options in which way to navigate and present this data. I showed the shared dysregulated proteins and the both up and down regulated. For this figure, we did delve into uniquely dysregulated. I wanted to also cover proteins or pathways in 2 out of the 3 synucleinopathies, because one of the interesting finds was that we had a huge overlap between DLB and MSA. A lot of research is focused on PD and DLB in particular. So this addition of the MSA showed that it is a vital component out of the 3 synucleinopathies and actually shares more proteomic changes to DLB rather than the PD itself.
Sorry. Do we have time for one short other question? Because otherwise we don't get to that one. So, which is on figure number 6, in which you present differentiating proteins. In panel B, you present the Z-score of 37 individual proteins. So, can you explain me how that Z-score was calculated and what it represents?
So for this analysis, we took out the controls and carried out an ANOVA between just the 3 synucleinopathies themselves. And this is where we identified these 37 proteins. In order for me to visually represent from the hierarchical clustering, I z-scored using Perseus. So I categorized it down to either -1, 0, or 1 to show either a positive or a negative intensity.
Okay, in view of time, I give word back to the pro-rector. Thank you.
Thank you very much. The opposition will then be continued by Dr. Wilma van den Berg, who has expertise in neurodegeneration, who works at the Amsterdam UMC and the Free University of Amsterdam.
Thank you very much. Dear candidate, first of all, I would like to congratulate you. you have brought a wealth of data to the field in which there's lots to learn from. And I think your thesis is very well written. It's really built up, and it's nicely brought in the context. So very well done. I also really like the images that you brought in to really understand what are the differences or how your workflow is. So I think that helps a lot of people to understand what you did and what we can learn from these data. So really nice. I would like to go first to chapter 2 with you. And what I saw is that you really brought together quite a large cohort to do proteomic analysis. This is quite important because, as you know from post-mortem analysis, and I'm a neuroanatomist by training, so I look at these brains a lot, we can see a lot of variability. And now, not only between the groups, MSA, DLB, and PD, what you studied, but even within those groups. So kind of my first question that has to do a little bit with this cohort and the groups that you put together for this analysis. Like one of the things that you saw, and you pointed out in figure 1C on page 41, is that there's no difference in the levels of alpha-synuclein in both the soluble and insoluble fraction. Could you explain that?
Thank you, highly esteemed opponent, for your kind words and for the question. So the changes, or the lack, thereof, a significant upregulation of synuclein in both the soluble and insoluble fractions is something we have observed several times. What we believe is it is not the overall amount of synuclein that is increasing, but rather the type of synuclein. So first of all, we think maybe there may be a balance between cell death, neuronal death, and the aggregates that occur in those cells, causing a restoration of normal synuclein also. And from our modification analysis, we can identify that although the amount of synuclein is not higher in the insoluble in the synucleinopathies, the type or the version of synuclein is different. So we see it's highly modified with both acetylations, ubiquitinations, and phosphorylations in total also.
Okay, that's exactly where I want to go to. So first, what you show is that you don't see a difference in synuclein. Actually, most cohorts have shown that there's a huge biological variability. Mm-hmm. So both in CSF and brain tissue, there's lots of variability in the amount of alpha-synuclein. And maybe the structure and modification matters more. Maybe you agree in this. I think there's lots of hints in your thesis that really point towards that. And there are several post-translation modifications that you already discussed on page 40. In controls, you see some acetylation, but in the synucleinopathies, you see multiple modification. Now, my question is, like, what is physiologically relevant? What is pathologically relevant? That's very difficult. And how would you study that in more detail?
So I agree with you. I think the modifications are a big impact and have the most probably definition of the pathological version of alpha-synuclein. However, we do not know what is a modification caused after aggregation or is the initial initiator of the aggregates themselves. So based on that, I believe we can do one possibility is single cell analysis or deep visual proteomics, where we would actually focus in on a cell in terms of single cell or an aggregate in terms of deep visual proteomics, and we would be able to assess the synuclein that is in that cell or in that aggregate exactly, and then look at a region that does not have aggregates present, maybe an earlier stage that has yet to have pathology, and do the same analysis. So look at the cells or areas using deep visual proteomics where there's no aggregate formed, and see what the synuclein looks like in the brains of those patients before an aggregate has occurred.
Mm-hmm. Yeah, so looking at an area that is more resilient compared to an area that shows a lot of pathology, and maybe more on cellular level. That's what you're saying.
Yes.
So on cellular level, and we did a lot of microscopy analysis, we actually do see quite some differences. One of the statements that you wrote down on 42 is that especially ubiquitination and phosphorylation, which is in my view, the most studied PTMs, post-translation modification, is actually correlates with pathology formation. I think that's a very interesting statement. Did you study that in more detail in your cohort? Because basically you have the tools to do so.
So one analysis in my third study where I did the MSA in particular cell model. For that, I used cerebellum tissue, where we saw an increase in the aggregates based on staining, but also I compared the proteomic analysis between the cerebellum and the cortex, and I did see that there was an increase in ubiquitination in the alpha-synuclein in the cerebellum, where also based on staining, we had increased aggregates, total synuclein, and PS129. So based on that, I do believe that the ubiquitination is a very important modification for pathology. We also identified an increased ubiquitination site in the cells when the inflammation was added, showing that where we saw more aggregates also correlated with an increase in ubiquitin sites, but also frequency in the cells that had inflammation also.
Very nice. I think we see ubiquitination early, and maybe other sites are later. One of the things that I missed kind of was truncation in your discussion. Do you think it's relevant?
I think it's very relevant for truncations and cleavages. I chose not to focus on those for the first comparative proteomic analysis. However, I did look into them for the cell model based on the fact that we could see a before and after, or with and without stressor or increased in aggregation. So with the inflammation, I saw that there was increased cleavage at the N and C terminal, and I think one site in the mid-domain versus the cells without aggregates, or even the cells with small amount of aggregates and no inflammation, where I saw mainly cleavage sites in the NAC domain. And as we know, the NAC domain is the main component, we believe, to be to make up the aggregate itself.
Okay. I would like to stop here, because I have a few more questions that I would like to address. One is related to the figure, actually, that we just discussed with the other opponent on page 72, in which you say, actually, the MSA and DLB proteome overlaps quite a lot. I was wondering whether that not has to do with the fact that you chose cortex instead of like MSA, oligodendrocytic pathology is mainly cerebellum and striatum, and the cortex, we mainly see Lewy bodies also at end-stage MSA. What's your view on this?
So for this comparative analysis, we focused on the cortex because all three diseases have pathology in the cortex, especially at the late stage where we are receiving the postmortem tissue. I do agree that it could be some sort of regional differences, of course, between looking at the cerebellum and the cortex of patients with MSA, which we did look at. We found that there was an increase in inflammatory markers in the cerebellum, the JAK-STAT pathway, HLAs. However, based on the comparative analysis, we wanted to focus on one area because we also looked at the substantia nigra of PD patients. However, we felt to really provide the field with a resource of interesting proteins and pathways, one region of interest would be more interesting.
Thank you very much. I'm going to cut it short here because I want to ask one last question, if I can. Very short question. You talk about the continuum between DLB and AD in chapter 3 and distinct profiles. One of the things that was posited is that you see co-occurrence and co-localization between tau and alpha-synuclein. Are you sure that it's within the same cell?
So for this analysis, we carried out both simple IHC and also double immunofluorescence. What we did observe is overlapping regions between the DAPI staining of the synuclein and the tau. However, there was also regions where there was purely aggregates of either tau or either synuclein. So whether they fully co-localize, we think there are areas of co-localization in these patients.
So it's co-occurrence within the same region?
Yes.
Okay, I agree on that comment.
Well, given the time, we have to move on to our online members of the committee. And we will give the word to Dr. Sasha Singh, who is a director of proteomics research at Harvard Medical School.
Dear candidate Greally, I want to echo the sentiments of the previous speakers that this was a beautifully written PhD thesis, especially the introduction For someone who's not in this field, I really appreciate the background. And I also appreciate the fact that you implemented very simple figures. Often proteomic studies or theses can overwhelm the reader with unnecessary networks and information that distract from the main message. My first question, so I have a couple of specifics and then I have one big picture question. Pertaining to chapter 2, actually, I was a little surprised about how you presented some of the data in your thesis, given that you're from the Steen Lab, particularly regarding your consensus proteomes and your consensus PTM profile. I'm curious how you actually generated those consensuses. Did you do a consensus proteome and PTM per cohort, or did you do it together? Because the more samples or the more diversity in your samples that you have to create consensuses, the more likely you have missing values across the different patients. So how did you actually create these consensuses? Because it relates to now my actual question.
Thank you, highly esteemed opponent, for your lovely words and your question. So for the PTM analysis, that was a present or not present analysis over all the DLB, PD, and MSA patient groups using Mascot and MSFragger. I combined both softwares for the PTM present or not present. I decided to present it as a cumulative map to simplify it, and also to not focus the main findings or the main message of my first paper or first study to be focused on synuclein itself. I wanted to emphasize, we wanted to look at the other proteins and other pathways of the whole proteome of patients with this synucleinopathy, and therefore that's why I decided to show a cumulative map for the first figure.
Okay, so let's talk about first the PTM profile. Unfortunately, again, as previous opponents already mentioned, I have no sense of diversity or heterogeneity across your different patients within a group. So how much does your figure 1 map, for example, represent the different cohorts? Like how prevalent or how frequent do you have these modifications? Or is it 1 or 2 patients that are driving this consensus?
Thank you.
And don't you think that matters? Yeah, don't you think that matters when we're talking about, between and within cohort heterogeneity?
Yes, thank you for your question. So for this analysis, focusing on the global proteome changes, I did find that the PTM analysis of the synuclein was more difficult in this cohort. I did identify some really interesting PTMs, in particular the PS129, which is a difficult PTM to identify via mass spec. However, it was in low frequency in my PD, DLB, and MSA group. And I think that is due to that we have yet to optimize and enrich for the pathological form of alpha-synuclein to carry out more of a targeted approach to study the strain or proteoform specific versions of synuclein. So this is something I want to focus on next, because I now have identified the molecular pathways and the other proteins that are dysregulated in PD, DLB, and MSA. The next step is, what is the strain specific type of synuclein present in PD, DLB, and MSA? And is that because of some of the pathways and proteins that we have identified? So something like a more targeted approach like the Flexi analysis, where we're looking at carrying out a Flexi Syn, where we've generated a heavy labeled alpha synuclein, where we spike this in to our brain tissue samples. And this enables us to look at the peptide level, the heavy labeled peptide versus the peptides or the endogenous peptides of our samples, and it enables us to see the modification extent or the changes between both sets of peptides, where we can then hone in on these regions of interest, knowing that there is a deviation caused by a modification or a cleavage.
Okay, so then that would suggest that your total alpha synuclein may actually be incorrect, because you haven't applied, between soluble and insoluble, you haven't applied that standard to confirm or account for those PTMs.
We haven't done any absolute quantification using something like the Flexi.
Another question about the PTMs I didn't understand. So you often refer to ubiquitination, but this would generate a KGG footprint. That's also present, correct me if I'm wrong, in netylation, acetylation. So how do you know for sure that these are ubiquitin sites and not some other homologue of a ubiquitin?
Thank you for your question. Based on my knowledge using the softwares like Mascot, MSVager, etc., we input the exact mass of ubiquitination, the 114, and this helps us then to identify the sites of interest. I'm not sure if that.
Well, let me ask you from another perspective. Did you ever identify ubiquitination in any of these? Often, if proteins or tissues heavily ubiquitinated, I know you didn't do pull down. Did you have evidence of ubiquitination in any actual ubiquitinated peptides in your samples?
Yes, we did identify overall ubiquitinated proteins or peptides. I also identified ubiquitin itself, the protein, being significantly upregulated in my DLB tau positive patients. So I did find the peptides that make up ubiquitin there also. And lastly, in my cell model, we applied a general FK2 ubiquitin antibody because there's no specific antibody for ubiquitinated synuclein. And we also saw an overlap and staining, positive staining for ubiquitin using this antibody in the cell model also.
Thank you. Going to the cell model, I have some questions about your description of some of these typical pro-inflammatory proteins, such as STAT1, such as the STATs. Do you think it's sufficient to see an increase in these proteins, or what about their regulation? For example, STAT1 works as a transcriptional regulator that, when activated, its PTM profile is modified. So did you look at any modifications of STAT1 or the different STATs like phosphorylation to know that it's actually activated as a true indication of inflammation?
Thank you for your question. We did not look at the modification of STAT. For this analysis, it was the significant upregulation of STAT1 and other JAK-STAT pathways that I identified. I agree that mass spectrometry cannot always tell us the activity of said protein. So all we know is it's increased or decreased in expression. However, yeah, looking at something like the modification is something I will take into consideration. Thank you.
In the light of time, I'm afraid I have to stop this discussion and move on to our final online opponent, which is Dr. Dominique Winter, who is an expert in proteomics and molecular biology and works at the Institute for Biochemistry and Molecular Biology at the University of Bonn.
Hello, thanks a lot for having me. And also, I'm a little bit redundant to my predecessors, but congratulations on a really nice thesis. And I think everybody working in the field knows how difficult it is to get so many patient samples, to make so many analyses, to get so many nice, coherent figures out of it, and to go all the way from patients over biomarkers to cell culture models. So yeah, very nice. Yeah, I really enjoyed your data and the wealth of data you produced in a PhD. That's really very nice. So I hope I don't introduce any redundance. I would like to briefly pick up on the PTMs as well, as already Sasha mentioned. And there I would have one question, because in several of your figures you mentioned the sequestosome, SQSTM1, as an upregulated protein. And what's your take on SQSTM1, and why is this always popping up? What is the connection to your insoluble alpha-synuclein?
Sorry, can you repeat the last part of the... Oh, sorry.
So the sequestosome, SQSTM1, it's like one of the top regulated. You also highlighted this in many figures. So what's your take on this protein with your aggregates?
Thank you, highly esteemed opponent, for your kind words and for the question. So we regularly saw the sequestosome, or P62, significantly upregulated in all synucleinopathies in both by proteomic analysis, but also staining analysis using antibodies. I believe that this is a well-known protein involved in many proteinopathies due to its role in the clearance or autophagy system and ubiquitination system. I think it is upregulated in all proteinopathy diseases, including Alzheimer's and the synucleinopathies also. It could be consequence of the aggregates themselves, causing an upregulation in which the neurons are trying to clear and get rid of the aggregates, or it could be having an impact on the aggregation process itself. This is still something we have yet to figure out.
So in light of the 3,000 proteins you find in the insoluble fraction, one could speculate that they are bound together by P62 because it binds to polyubiquitinated aggregating proteins. So what's your take on that? Do you find ubiquitination sites also on the other proteins which you enrich in these fractions? And do you think they are synuclein specific or P62 specific?
Thank you for your question. We did identify other ubiquitinated proteins and also more aggregate-prone proteins in our insoluble fraction, like tau, Huntington, etc. Any protein we identified, I also assessed its level in the soluble fraction. Whether the increased ubiquitination is due to P62 or the sequestosome, this is something we have been interested in. Looking at a cell model in particular, I have assessed some different stressors, proteasomal stressors, increasing ubiquitination, etc. And this has been assessed by cell models. However, I did find that the inflammation was the pathway that had caused the increased aggregation out of all stressors applied.
But what's your gut feeling? Are these studies of P62 of which synuclein is a part of, or is synuclein the seed to generate the aggregate? Because these are two different fundamental questions, right? So what would you think after all these analyses?
I think we would need additional analysis on brain tissue specific. So instead of carrying out something like brain homogenization and sarcosyl, we could do something like cross-linking mass spectrometry, where we would look at the interaction between the P62 or sequestosome and alpha-synuclein to see if they do interact.
Very politically spoken, like a true politician. Another thing I missed a little bit in your thesis is that, especially in Parkinson's disease, organellar dysfunction has been a hot topic for many years. Mitochondrial dysfunction, lysosomal dysfunction, GBA1 is the highest genetic risk factor, it's a lysosomal protein. So did you find any patterns in your large-scale analyses pointing towards organelle-specific phenotypes other than VDAC1, which you mentioned?
From my MSA analysis in particular, we saw a most significant upregulation of mitochondrial associated proteins like the VDAC proteins, SPART. So those proteins definitely point to an organelle-specific dysregulation in the MSA patients. However, of course, something like more single-cell analysis or something on a cellular level would give us more information on the different organelle dysfunction.
Okay. And maybe to briefly stick to the organellar influence, so in your cell culture model, you give these seeding lysates from brain to HEX cells, right? And I'm not an expert in this model, but somehow the cells need to take up these proteins, and this should be through the endocytic pathway. So how can it be that the protein entering the cell through the endocytic pathway results in intracellular aggregates, which is stained later by immunofluorescence? So how should this work? Are these intra-endosomal, intralysosomal aggregates? Does it magically transition to the cytosol? Are they extracellular? What's your take on these mechanisms you investigated there?
Yes, that's a really good question. So for the cell model, we use lipofectamine, which uses lipid droplets to transfect our insoluble fraction into the cells. What we did observe was the addition of the MSA seeds, that we call it, is needed for this aggregating process. So the synuclein itself, that we believe is pathogenic from the insoluble fraction from the MSA patients, was needed in addition to the inflammation to cause these puncta or aggregates of alpha-synuclein. When we applied the inflammation alone, we did not see an increase in puncta or aggregates. So we believe it's the combination of the two of them together is needed.
All right. I guess I have 1 minute left, or one last question? Or
No, the hour has passed now. The candidates...
Then thank you very much for this nice answer.
I presume you finished your answer as well. So, Sinead Greally, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. The mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Sinead Greally, the degree committee here present, has assessed the quality of your thesis and your defense. In view of its positive verdict, and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Blokland is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
I will.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Sinead Greally, the degree of doctor and grant you all rights attached to custom and law. As evidence of this, I will now present a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Congratulations,
You want to take a picture? No, you're
Oh, yeah. Sorry. Thought we had to.
Okay. Ik wil plaatsen. Before I give the word to Judith for the laudatio, I first would like to thank Jan Ramak, especially because he stepped in for Wim Riedel. So very much thanks for doing that. And of course, the people who made it possible to make this defense take place here. We have Theo de Kok, Maarten Honing, Wilma van den Berg, Sasha Singh, and Dominique Winter. Thank you very much. And of course, also to remember that you're one of the first research master students from Maastricht, and that you now, well, in full glory, shine here today. So wonderful achievement. And now, on behalf of our research master of drug development and neurohealth, and I'll give the word to Judith. More funny,
Thank you.
interesting things.
So, I am so proud. I think you really stepped up, and I just wanted to say thank you to everyone here for showing us how good Sinead was or is. You know, I think you've grown incredibly since you came. I'd like to thank Rudy for bringing her to our lab. It takes, as I always say, it takes a village to raise a scientist. And Maastricht has been a big part of this raising of a scientist and bringing you to this level. So I'm very grateful to the program and for connecting me with you. I have to say, you know, initially Sinead was doing a project at Pfizer, and then Pfizer shut down Neuroscience in Boston, and so we had this incredible opportunity to take Sinead on. And at first I was a bit worried because Sinead had, you know, I had asked her, what have you done before? And she said, well, I've done some Western blots on synuclein. So I thought, she hasn't really worked much with proteins or wet lab, but people can learn. So she did. She really learned a lot. And, you know, along the way, we had a lot of help from other people in the lab. We have a community of scientists in our lab who have different expertise. And Sinead has been one of these connectors. If she needs something when she's doing her work, she'll go and find the expertise in the lab. and try to talk to the people who have the expertise and gain the expertise from them. She has a very thick skin. You know, she worked with all of the people in the lab, and you know, irrespective of personalities, Sinead can get things done and learn from everybody, and I'm so incredibly proud of that. You know, there were several people in our lab who worked with her, like Long Cheng, who taught her a lot about the cell cultures. Dr. Mukesh Kumar, so Dr. Long Cheng, Dr. Mukesh Kumar, and she's been incredible with respect to helping us with interviews of new students coming in or postdocs. She has a very good eye for good people. So I always listen to what Sinead says. So I'll have you know that you've had several PhD offers or postdoc offers from other members of the committee. So, you know, in all, I'm grateful to Ariane, Rudy, Hanno, and all of the others in our lab, our colleagues who have helped me to raise you as a scientist, and I'm incredibly proud. And thank you to all of the opponents. You really brought the best out of Sinead today.
Thank you.
Dear Dr. Greally, also on behalf of the Faculty of Psychology and Neuroscience and Maastricht University, I congratulate you with the degree that you have acquired. So we're almost done with the formal part of this ceremony. I would just like to mention that after we close, we will gather just in front of the screen here so that there will be an opportunity to make a picture of you with the entire committee, including our online members. After that, we will have another photo session in the hallway just in front of the stairways. And with that, I close this ceremony. Thank you very much.
Paan, if you can...